We need some fundamental components to do real-time PCR. We need a real-time instrument. It does have a thermal cycler, but also has an optical detection system. We also need a real-time PCR fluorescent chemistry. So the chemistry will emit a fluorescent signal automatically with each cycle. Real-time PCR is particularly effective for gene quantification due to the ability to detect the exponential, or if you prefer, geometric or logarithmic phase of PCR. Exponential phase efficiency is very consistent, so the original quantitative relationships of genes across samples are maintained throughout that phase. So with real-time PCR, we're collecting typically 40 cycles of data, although you can adjust that if you like. But we're only going to select one special data point from each amplification plot at a consistent point, and that point is in the exponential phase. Those special data points are referred to as CT values. Now, if the goal of the assay or test is to quantify, we can calculate the initial target quantity by using the CT value and the exponential PCR phase efficiency. It's based on the relationship that you see here. The quantity is proportional to the exponential efficiency, which is the E there, taken to the minus CT power. So this fundamental relationship is the basis of the real-time gene quantification method called delta-delta CT, or sometimes called comparative CT. This is the best method known for quantifying genes in real-time PCR. For those who have gel-based experiences with PCR, then it comes as no surprise, I'm sure, that you realize that PCR routinely amplifies non-targets. And we can detect that on gels when you see bands or smears that have molecular weights that don't correspond to the target. But one of the benefits of gel electrophoresis is that we can physically separate target and non-target if they have a different molecular weight. But here's the key difference. With real-time PCR, there is no physical separation of target and non-target. Everything stays inside the tube. There are no gels inside the real-time instrument. So this presents a challenge to all of us users. We need a way to generate signal from the real-time PCR reaction that only comes from target, despite the fact that PCR routinely amplifies non-target. So the first and still today the gold standard real-time PCR chemistry is the 5' nuclease assay, more commonly referred to as TaqMan. The 5' nuclease assay gets its name from the 5' nuclease activity of Taq DNA polymerase, which I've rather simplistically drawn as these two colored spheres. The green sphere represents the 5' nuclease domain of the protein. So during DNA synthesis, this nuclease domain can cleave or clip DNA that may be bound downstream of DNA synthesis. So think of it as the nuclease activity is removing these strands in anticipation of upcoming synthesis. 5' nuclease assay also utilizes a technology called FRET, fluorescent resonant energy transfer. This was a phenomenon discovered years ago by a German scientist named Förster. Now we'll start on the left. Supposing that we have two fluorescent dyes, a green dye and a red dye. You may be aware that green light has a shorter wavelength compared to red light. The shorter the wavelength, the higher the energy. So the green dye is considered a higher energy emitter. So you might think if you excite the green dye, typically done with blue light, you'd expect to see a strong green emission signal. And normally you would. But Förster discovered is if you brought this red dye in close proximity, now when he stimulates the green dye, the energy of emission transfers through space to the dye that emits at the lower level, in this case, the red dye. So think of it like water flowing downhill. Energy likes to go from a higher energy state to a lower energy state. So on the left, therefore, we could say the red dye is absorbing or quenching the signal from the green dye. Quenching being the technical term. But FRET depends on the distance between the dyes. So if we separate the dyes in space, as we imagine on the right, then FRET does not occur. So if we stimulate the green dye it gives us the strong green signal that we would expect. So we put this all together into a nuclease assay, and this could be for lots of different applications, gene expression, microRNA, RNA viruses, all sorts of things. The nuclease assay would typically consist of two PCR primers, which are unlabeled, and there's a third oligo called the TaqMan probe that is labeled. It's labeled on the 5' end with a dye that we call the reporter. I want to make sure I saw a message up here. See if there's anything going on. See anything going on? Okay, sorry, just checking because I saw a little message pop up. The 5' end has the reporter dye, and it's called reporter because the signal from that dye will report on the amplification of the target sequence. On the 3' end is a quencher molecule, which, of course, you can tell from the name, is going to quench the reporter. But in addition, it's going to block the 3' hydroxyl on that probe, so that way the probe cannot be extended like a primer. And you can probably tell from the drawing, the probe is gene-specific, designed to bind between the two primers. Now, initially, when you're making an assay master mix, so we've got primers and probes floating around in solution, the probe is intact. It's rather flexible. So when the probe is intact, the reporter and the quencher have a natural affinity. They come in close contact. So if you were to measure the reporter signal at this point, before PCR, you would find it to be very low. It is being quenched. Next, we hit start on the real-time machine. Eventually, it will reach the annealing temperature. This allows the primers in the probe to anneal to the template strand. Now, if we can just imagine that very split second when the probe has annealed to the template, you see in this drawing, it's stretched out in double strand configuration. The distance between the reporter and the quencher is too large to allow for it to occur. Theoretically, I suppose, if you had a super fast, super sensitive camera, maybe you could pick up that initial moment when the probe becomes double-stranded. But, as I said, the camera would have to be amazingly fast because in the next, say, microsecond, TAC extends from the upstream primer and comes in contact with the 5' end of the probe. Then a key event occurs. If the probe is bound to the correct target sequence, TAC's 5' nucleus activity can cleave it. This generates a fragment containing the reporter dye, which just floats away. So at this point, immediately following cleavage, the reporter and the quencher are on separate molecules. So that reporter that's now free will never again get quenched during PCR. So we can see that this probe is now two pieces. We've got the reporter fragment, the quencher fragment. They won't participate in PCR any longer, but they'll act as permanent records of amplification. Now, if you remember, a few moments ago, I showed a picture of a gel, PCR gel, and you could tell that there were some non-target products there because they were of different molecular weight than the target band. So that was meant to introduce the concept of specificity. So assay specificity can be defined as the degree to which the assay includes target and excludes non-target from the results. What I've done in this slide is represent specificity in a kind of symbolic, or you could say mathematical way. So the green bar represents target molecules in your sample, red bar represents non-targets. And in the final result, what we want is if there's any contribution to that result by non-target, we would want to be mathematically insignificant, at least for gene expression, realizing that there are some applications where you can't tolerate any non-target contribution to the results. But at least in things like gene expression, microRNA, we might be able to tolerate a little bit. So when thinking about specificity, it might be a good idea to think about what is our greatest threat to specificity, because if we can handle that threat, lesser threats are easier. So the greatest threat when it comes to TaqMan to specificity are homologs. The homologs are genes which have a sequence relationship to the target, but it's not the same gene. We don't want to pick it up, we don't want to detect it, but they're there in your sample anyway, because homologs are extremely common. They can be found easily within a species, but also even across species in many cases. So how would TaqMan probes help us to exclude homologs from our results? Which, by the way, homologs have been a problem with gel-based PCR for many years. Pardon me. So let's imagine that we're doing, it could be gene expression, it could be microRNA, what have you, and there's a homolog present in your sample. So the homolog, let's say, has a partial primer binding site. So there are mismatches in the primer binding site. The mismatches don't mean the primers can't bind, they can. And if the primer is bound to the homolog with the mismatches, and there's a nearby Taq polymerase, the polymerase can bind to the 3' hydroxyl of that primer. Once that happens, we have template, the homolog, we have the polymerase bound to 3' hydroxyl, so it's ready to go. We've got the NTPs, magnesium chloride, a good temperature. Everything is there to allow for DNA synthesis. So Taq extends from the mismatched primer. When it extends, it's also stabilizing that mismatch. Now, Taq is pretty fast polymerase. So if, let's say, I'll just pick a number, let's say there were 6 base mismatches in the primer binding site. Taq could extend 6 bases in just a fraction of a second. At that point, the primer is now bound to the homolog just as stably as if it had been bound to target. So at that point, there's nothing to stop Taq from completing synthesis of the homolog. So what I'm showing here, what I'm representing here, is a phenomenon that was published many years ago called primer mismatch extension. And it explains why it is that even though the researcher didn't want to amplify the homolog, it happened anyway. If you think about it, it actually makes perfect sense. Because with primers, we're demanding 2 functions, but those functions are oppositional. On the one hand, we're certainly hoping for specificity. We always want that. But at the same time, we demand that primers provide a 3' hydroxyl, and that hydroxyl is an opportunity for the polymerase to extend a mismatched primer and stabilize it. So you see how those 2 functions are fighting each other. That's just the nature of primers. That's the way that works. Okay, so we can say then that Taq polymerase can mitigate the effect of mismatches in a primer binding site. Now the question is, can Taq do the same thing to a probe? The answer? No. Why? Because the 3' of the probe is blocked by the quencher. Well, that's good news for all of us, because that means whatever those mismatches are in the probe binding site, they're going to have their impact. Taq can't mitigate that. So what happens? Taq comes along, hits the mismatched probe. Now, because of those mismatches, the melting temperature, or TM, of the probe has been reduced. It's been reduced to the point where it can no longer be cleaved by the polymerase. So instead of being cleaved, the probe is displaced from the homolog, where it instantly recoils back to its original quenched configuration. So that means, even though homolog amplified, you didn't generate any signal from it. Therefore, your data remains clean. So I would argue this is the big philosophy behind TaqMan. We're saying with TaqMan, it's very difficult to prevent all non-targets from amplifying during PCR, but through the use of a well-designed probe, we can prevent those non-targets from producing signal. The other type of non-target that PCR very often generates are called non-specific products. So in a sense, these are the opposite of homologs. These are sequences that are unrelated to the target. They're formed seemingly randomly. So you could have primers bind to some random sequence in your sample, and they just, by chance, are in the right orientation to produce a PCR product. Some people refer to these as primer dimers. Well, good news, because by definition, these non-specific products do not have a sequence relationship to the target. There's nowhere for a TaqMan probe to bind to a primer dimer, much less get cleaved. So we have automatic exclusion of all non-specific products from our TaqMan results. That's actually a huge factor. When our company measured the specificity of TaqMan assays, it could be done on an industrial level because of this fact. Had this fact not been true, we could not have done that. Well, with that little introduction to real-time PCR, I'd like to get into one of the main topics here, microRNA. So microRNAs are short, single-strand RNA molecules that are powerful regulators of gene expression. In the case of animal cells, it's likely that the microRNA will bind to the targeted messenger RNA with a mismatch. This will result in translational repression. In the case of plants, it's more likely the microRNA would bind to the targeted messenger RNA with a match. This results in cleavage and subsequent degradation of the targeted messenger RNA. MicroRNA is produced through a series of processing events. So we have an initial transcription producing what's called the primary or pri microRNA, which has a series of hairpin loops. Next, this primary RNA is processed into what's called the precursor or pre-microRNA, which is a single hairpin loop structure, which is then exported to the cytoplasm, where it undergoes additional processing into the mature form, single-strand form. The single-strand mature form will then interact with what's called the RISC complex. This complex is now biologically active and can act against the targeted messenger RNA. So let's say you're interested in microRNA experiments. Perhaps the first step might be, how are you going to extract the RNA? Because there are some RNA extraction methods that are not designed to pick up very small RNA molecules. So that's a very important point. Make sure you select an RNA extraction method that will do that. Some examples here are called Mirvana, and there's different versions of Mirvana. There's the glass fiber filter method and magnetic bead-based separation method. Another key point about microRNAs is specificity. So most of the microRNA research is directed towards the mature form because that's the form that's biologically active. Mature microRNAs are particularly challenging targets for specificity. First of all, we have to be able to exclude the earlier forms, the primary, the precursor, from our results, if we're just focused on the mature form. In addition, the mature form is very short. I mean, how do you find enough room for 2 primers in a probe? That's a challenge. And many of these microRNA genes only differ by, say, 1 or 2 bases. So that presents a significant specificity challenge. So one way to address this challenge is illustrated by our TACMAN microRNA assays. So first, we start with a hairpin loop reverse transcription primer. So if you can imagine the very right tip of the blue mature microRNA drawing, that has the 3' hydroxyl. That 3' hydroxyl is only available in the mature microRNA, not in the precursor, not in the primary. So therefore, the hairpin loop reverse transcription primer can only stably bind to this mature form. So we're getting specificity right from the very beginning, from reverse transcription. Following reverse transcription, the cDNA is transferred to the real-time PCR reaction. The hairpin loop unfolds. This increases the size of the target sequence. This allows us to have forward primer, reverse primer, and a TACMAN MGB probe. And we, as I said earlier, we measure the specificity of our assays, and the most challenging ones we find are in the LET7 family. So this is showing the specificity measurements that were done. Specificity measurements in this case for microRNA, we're using synthetic RNA molecules. The reason for that is that we are using gene-specific reverse transcription primers, so therefore some of the specificity is happening during reverse transcription. So for the TACMAN microRNA assays, we recommend the TACMAN microRNA reverse transcription kit. This kit does not contain primers, which is often different from a lot of other reverse transcription kits, for the reason that the reverse transcription primer comes with the TACMAN microRNA assay. Again, it's a gene-specific reverse transcription primer. Now we offer another chemistry for microRNAs called TACMAN Advanced microRNA assays. And there's a number of steps here. So we start with a polyadenylation step. So we're adding poly-A tail to the 3' end of the mature microRNA. Then an adapter is ligated to the 5' end of the mature microRNA. Then we're going to perform reverse transcription using a universal reverse transcription primer, which of course includes oligo-DT. The cDNA is then transferred into a chemistry called MIR-AMP. The MIR-AMP chemistry has a forward primer, which matches the adapter sequence, and a reverse primer matching the universal primer sequence. And we amplify that 14 cycles. The MIR-AMPed cDNA can then be transferred over to real-time PCR. So the reverse transcription kit, and actually there's more going on here than reverse transcription, is called the TaqMan Advanced microRNA Reverse Transcription Kit. And you can see a lot of components because there's a lot of different reactions going on there. But this is one we'd recommend for the advanced microRNA assays. So let's compare the TaqMan microRNA assays to the TaqMan advanced microRNA assays. So the microRNA assays use gene-specific priming, as I said before, whereas advanced uses universal priming for reverse transcription. The TaqMan microRNA assays use fewer steps in the procedure. Advanced uses more steps and takes naturally more time too. The TaqMan microRNA assays, that chemistry is compatible with SNO RNA. Many researchers doing microRNA experiments will use small nuclear RNA as their normalizer. Not saying you have to. I'll talk about that a little bit later on. Whereas the advanced microRNA assay chemistry is not compatible with SNO RNA. So whether you made cDNA from the TaqMan microRNA assays or from the advanced MIR-AMP product, either way, that would be transferred to real-time PCR with the microRNA assay of interest and TaqMan Master Mix. We recommend the TaqMan Fast Advanced Master Mix. From the name, you can tell it can be used in fast mode. That's fine. Or you can use it in standard mode. So currently we have over 5,000 advanced microRNA assays, over 22,000 microRNA assays, and over 3,000 primary microRNA assays. So the next question is, let's say you're interested in microRNA research, you'd like to obtain some microRNA assays. So one way to do that is go to our Thermo Fisher website, and the default search is search all. What I'd like to do is change that to TaqMan assays. It's very simple to do. Next, we recommend for the search parameter to use the mature microRNA sequence. A reason for that is that the nomenclature for various microRNAs can differ from database to database, but the mature sequence there's no ambiguity there. So that's what we recommend for the search. Very often you may find multiple assays for that particular sequence. As you see here, we have an advanced assay, one TaqMan microRNA assay, and 7 primaries. So choose the assay type that you wish. In this case, chose the TaqMan microRNA assay. The default size of the assay is small. There's a little S, I don't know if you see that there on the right. But we can change that. We have different sizes. And it's showing you that you're going to get 2 tubes. There's the RT, that's the reverse transcription primer tube, and then the PCR, that's the 2 primers and TaqMan probe tube. Also, we provide a lot of bioinformatics information about our assays, including the genomic map, which might be helpful. Additional bioinformatics information is microRNA sequences are often highly conserved across species, so it's showing that this particular microRNA is compatible with quite a few species here. But what if we don't have the microRNA assay that you want? Well, we have a solution for that. We have a tool on our website called the Custom TaqMan Small RNA Assay Design Tool. So it's web-based, and the tool will design for you uh, the hairpin loop reverse transcription primer, and the assay with the two primers in the TaqMan MGB probe. When a researcher's wanting to detect or quantify a small number of targets, then the individual tube assay format makes perfect sense. The individual tube assay is a concentrate, primers and probe. 20x is a very common concentrate, so it's ready to use, makes it very simple. But what if you don't know which microRNA targets are of greatest interest to the system that you're investigating? So in that case, you might want to consider an array. So an array can be defined as a PCR vessel of some sort where assays are dried down in specified well positions. So that way you can interrogate many targets per sample simultaneously. Arrays, for example, one way that you could use them is if you want to look at what, let's say, hypothetically, what microRNAs are of greatest interest for a particular metabolic pathway or disease. And we can test hundreds of microRNA genes all at once. So one type of real-time array is called the TaqMan array cards. The cards have the same size as a regular plate, but it's not a plate. It's not with open wells and you have to pipette individually. It doesn't work like that. It's actually simpler than that. So there are 384 microwells. Each well is 1 microliter approximately in volume. And they're initially connected with microchannels. So you would take one or more samples. You could have up to 8 samples, depending on the format of your card. And you'd have sample, like cDNA, for example, mixed with master mix, and apply that to fill ports. So on the left, I don't know if you see this, this is a picture of one of the cards loading the sample master mix mixture. Not really. They put red dye in there, but that way you can see what's being loaded. Following loading of the card, it's briefly centrifuged. The card is then sealed in like a 1 second step. The fill ports are cut off with a pair of scissors, and now the card is ready for use. And there are certain instruments where we can run the card, such as the QuantStudio 7, the 7 Pro, the QuantStudio 12, and so on. So we offer pre-configured microRNA arrays in the card format for human and rodent. And they come in a set, so there's A and B. The A card has the microRNAs that are most prevalent, shall we say. And there are 380 assays per card. Now, if you remember, with the TaqMan microRNA assays, we're using gene-specific reverse transcription priming. So I don't think anybody would want to set up 380 reverse transcriptions, but that's okay, you don't have to. We have pre-prepared reverse transcription primer pools. They're called megaplex primer pools. And they're available for A and B cards. So that way you only have to do one reverse transcription per sample per card. So let's say that you've made some cDNA and you're going to apply it to a card. So that cDNA will be divided among 384 microwells. But what if a certain microRNA has extremely low expression? So you only have, who knows, maybe 10 molecules or something of the cDNA. Then there's a chance that the cDNA for a particular target won't reach the well that has the assay for that target. So to keep that from happening, we have a chemistry called pre-amplification, which will increase the amount of each microRNA target quantitatively. So that way, even if you have a very low expressor, then some cDNA from that low expressor will end up in the assay that's designed for that target. So the way this works is, so you set up a pre-amplification reaction, and again, we have the preamp primer pools for the microRNAs. They're also called megaplex. So again, we've got two megaplex types. There's the megaplex reverse transcription primer pools. We also have the megaplex preamp primers. which match up with cards A and B for human or rodent. So you'd have the megaplex preamp primer pool, the cDNA that you made, and TACMan preamp master mix. So we're running only 12 cycles for preamplification. That ensures that all the microRNA targets are remaining in the exponential phase, and therefore their original quantitative relationships are being maintained. Then we dilute the preamp product. So the end result is that the concentration of each target, microRNA target, is increased by 100-fold compared to the original cDNA. So that guarantees that the microRNA, even if it's very low expressed, there'll be plenty of molecules. It should reach the well that contains the assay for that target. Now, the preamplification chemistry only applies to our, I'll call it the traditional microRNA assays, remembering that the advanced microRNA assays have a mandatory version of that preamplification called the MIR-amp. So preamplification does not apply to the advanced microRNA assays, is what I'm trying to say. And we do have advanced, in other words, cards that have the advanced microRNA assays dried down. And again, we're using universal transcription, reverse transcription priming for the advanced assays rather than the megaplex RT primer pools. Now, we also have another option for arrays called open array. So an open array is a specialized PCR vessel where we have over 3,000 microwells per open array. Amazingly, each well is only 33 nanoliters, which is so you're not going to use very much master mix, I can tell you that. But don't worry, it's not like you have to pipette 3,000 wells by yourself. No, that doesn't have that work. Instead, there's a robot called the AccuFill that will fill up your open array in about, say, 60 seconds or so. And so we have open arrays for our traditional microRNA panels for human and rodent. So each panel would have either 770 microRNA assays or 766 in the case of rodent. And we also have the TACMan open array human advanced microRNA panel. So now we're using the advanced assays in this case. Now, switching gears, many of you perhaps have heard of small interfering RNA or siRNA. These are short single-strand RNA molecules that operate in a way very similar to microRNA. So the siRNA would bind to the targeted messenger RNA, resulting in cleavage and subsequent degradation of that message. There are many experiments involving artificial siRNA added to cells, and the effect on the expression of a particular gene that's being targeted can be determined through real-time PCR. It's usually a time-based effect, as you can see in this simulated graph here. But what if you want to quantify the siRNA that's inside the cells? That can also be done with real-time PCR. We would recommend the TACMan small custom RNA assay design tool for that. It would design the hairpin loop reverse transcription primer, just like microRNA, and the 2 primers and probe for the real-time assay. Another type of small RNA is called the Peewee interacting RNA or piRNA. Again, these are short single-strand RNA molecules. They're thought to silence expression from transposable elements. Now, piRNA are slightly longer than microRNAs, but that's fine. They would still qualify as small RNAs. So if one wanted to quantify piRNA, that again could be done with real-time PCR, and we recommend the TACMan small custom RNA assay design tool. Some of you may know that piRNAs have on their 3' end O-methylated number 2 ribose carbon. Good news here is that this O-methylation does not interfere with the reverse transcription hairpin loop primer. And again, for these types of experiments, the sRNA, the piRNA, where you have the custom TaqMan small RNA assay, we'd recommend the TaqMan microRNA reverse transcription kit, just like we do with microRNA. A question that often comes up in regards to microRNAs is normalizing the data. So what that means is that when an experiment involves quantifying a gene in biological samples, cells, tissues, things like that, what we're really trying to do is to calculate the concentration of that target in that sample. So you think about, for example, gene expression. If expression goes up, the concentration of the transcript is going up. So to calculate concentration, we need two pieces of information. We need to know how much target was in the sample, but we also need to know how big the sample was the target came from. Together, that enables us to calculate target concentration. But typically, researchers will use a gene to normalize their target data by sample mass. So in other words, the normalizer gene is being used to measure sample mass. Some people use the term control gene, housekeeping gene. I like to use the term normalizer gene, but it's all the same idea. Uh, for those who are familiar with gene expression, you may have heard of genes like 18S, beta-actin, GAPDH being commonly used to normalize for gene expression experiments. For example, there's a lot of literature saying that the ratio of ribosomal RNA to total RNA remains fixed in cells across a wide variety of tissues, wide variety of metabolic circumstances, and so on. So as long as the normalizer gene is measuring the desired parameter, let's say total RNA, then that's a good gene to use as a normalizer. In the literature, that we see a lot of microRNA normalizing genes being small nuclear RNAs or SNO RNAs. The reason for choosing such an RNA it's not very clear to me. Um, my suggestion is, number one, figure out what is the normalizing parameter that you want to normalize your data to. Is it total RNA mass? Is it volume? What is it? What is most biologically relevant? If it is RNA mass, total cellular RNA mass, then there's no reason that you couldn't use a traditional normalizer assay, let's say for gene expression, to measure that mass. So if one feels compelled to use SNO RNAs, then I would suggest to generate data that shows that the SNO RNA measurement is an accurate measure of total RNA mass, assuming that that is your normalization parameter. Switching gears a bit, so let's supposing that you're doing cell culture in, let's say 96 well, they even have 384 well cell culture plates. The idea of extracting 96 different wells of cells individually is just a bit much. Well, in that case, you might want to consider, instead of extracting, to lyse the cells. It can be much easier, lower labor to do it that way. And we have a family of cell lysis kits called cells to CT, and they come in different varieties. We have one for gene expression, we have single cell, and microRNA. So the way it works for the cells to CT kits is you would, of course, remove the medium from your cells, rinse them out with PBS, and then add the lysis buffer, which contains DNase, incubate 5 minutes. Then add a stop solution, wait 2 minutes. So the incubation time in total, 7 minutes. That lysate can then directly be used in reverse transcription. But key to point out that we're recommending no more than 100,000 cells per lysis. Another cell lysis chemistry we have is called the superscript 4 cells direct cDNA synthesis kit. So in this case, the recommended number of cells to be lysed would be between 1 and 10,000. They are recommending that doing cell counting before lysis If you add more cells than the maximum, 10,000 in this case, then you're going to be exceeding dynamic range, and that's going to cause problems. So in this case, what they're recommending is trypsinize the cells, spit them down, rinse them out, add the lysis buffer, and then you can do reverse transcription. I had mentioned the term dynamic range. Might be a good time to say a few words about this. So dynamic range can be defined as the minimum maximum sample amount that may be run in the assay system and produce accurate and precise results. Generally speaking, in real-time PCR, maximum limit in your dynamic range will be defined by inhibitor. So in other words, if you add too much sample, you're going to run into an inhibitor that's going to create errors in your data, and you don't want that. The bottom boundary is based on what is the minimum amount of sample that you consider to be adequate. Because if you have too little sample, the sensitivity of your assay goes down. It may reach a point where it's not considered acceptable. So researchers create unique real-time PCR assay systems. So selecting a sample type, selecting an extraction method. If the target is RNA, reverse transcription chemistry must be selected, the volume of the chemistry, how much RNA goes into that chemistry, and on it goes. So there's lots of decisions being made. This creates a unique system and will have a unique dynamic range. Therefore, the dynamic range should be tested prior to proceeding to data production. So dynamic range testing, if the target is RNA, dynamic range test would involve serially diluting a representative RNA sample, creating an RNA standard curve. I'm saying one, but maybe in some cases you might need to try more than one. So each of these RNA concentrations are, of course, going to be reverse transcribed at some point. But a key point here is serially diluting the RNA, not the cDNA. That's a very key point. And I want to illustrate how important that point is with a real-life customer story. So in this situation, a customer was thinking about doing a gene expression experiment and thought to do a cDNA standard curve. So he made cDNA, serially diluted it. The cDNA standard curves looked fine, so it gave him the confidence to move on into data production. But then when the customer calculated the variability of his biogroups, so typically gene expression experiments, the samples are divided into biogroups, controlled, treated, things like that. And the variation in his biogroups seemed really excessive, so he was concerned. That's when he reached out to his local FAS. So there was a discussion with the FAS. The FAS suggested, well, maybe there was a problem in reverse transcription. The customer said, well, I don't think so because my standard curves look great. FAS says, yeah, they do look great. What are they made of? They were, of course, cDNA standard curves. The problem with cDNA standard curves is they're uninformative and they will typically always look good, which basically is fooling the researcher. So, for example, there could be significant problems in reverse transcription going on, but yet you're still making cDNA. Now, once you've made the cDNA and you serially dilute it, you're just propagating the error. So that's why cDNA standard curves always look good. It's a general rule. The right way to test dynamic range is not to serially dilute cDNA, to serially dilute RNA. So let's say one of the problems that could happen is reverse transcription inhibitor in the RNA. So that would present as what I like to call the tailing effect in the standard curve. So in this example, the highest RNA concentration reverse transcribed is shown on the right. And this is hypothetical, by the way. And if we look at the diluted, the very dilute points of the standard curve, we extend that out, that linear relationship out, we can see that the first point of the standard curve is elevated quite a bit, actually. If the RNA is diluted, that also dilutes the inhibitor. So the inhibitory effect should be reduced. And eventually, as you keep diluting the RNA, in theory, you should reach a linear response between the CT and the log of RNA reverse transcribed. And that's called linear dynamic range. So the researcher did that dynamic range test, and sure enough, it revealed the presence of reverse transcription inhibitor. So you can see that both for the target gene and the normalizer gene, the CT values were elevated over on the right. When the RNA is diluted, the second point from the right is elevated, not as much, because the dilution helped reduce the inhibitor. By the third point, third from the right, now we've reached the linear response between the CT and log of RNA. But you can tell that the samples were initially reverse transcribed outside of dynamic range, so that ruined the data. Now, some might say, but I'm using a normalizer gene. So I figure if there's an inhibitor, it's going to affect both the target and normalizer equally. So when I normalize the normalizer, that error goes away. Well, that's a lovely thought. Question is it real? Is it true? And you can see in this example, it's not true. So I calculated the RQ values within this range, in other words, outside dynamic range. And I came up with additional errors in the range of 10 to 15 fold. Okay, that's a lot of variation. Consider most researchers, when I talk to them and ask, what is your fold discrimination objective for gene expression experiments? The most common answer I get back is twofold. That is, they want to be able to discriminate a twofold change in gene expression. But if you're getting errors in the range of 10 to 15 fold, that blows that away. The data has been randomized. Data is no longer valuable. But there's no way to know that without testing like this, because gene expression data is essentially random numbers. Now realize this 10 to 15 fold, that's relative quantities. That's after normalization. So yes, both the target and normalizer were impacted by the inhibitor, but not to the same degree. That's where the 10 to 15 fold errors came from. Now, in the case of these cell lysis chemistries, the vendor should tell you the maximum number of cells that the chemistry will take for lysis. But just a point on that is that those tests were likely done on well-established immortalized cell lines. If you're working with other kinds of cell lines, like primary cell lines, you may want to do a dynamic range test by doing a cell dilution or cell standard curve test. So once you've gotten past the dynamic range testing, everything looks good there, now you're ready to quantify. And delta CT is the best method for real-time quantification. Now, in just about every case I've seen, microRNA experiments are relative in nature. In other words, relative quantification. We're not determining the number of molecules of microRNA. Relative allows sample-to-sample comparison of the same gene, but not gene-to-gene comparison for quantification. To do that, you would need absolute quantification. That is where we're determining the molecular concentration of the target in the samples. That would allow you to compare target-to-target abundances. In order to do that in real-time PCR, you would need an absolute calibrator. In other words, a sample where you know the number of molecules of the target, or the concentration, you could say, of the target in that sample. And there are ways to make those. So microRNA quantification data can be analyzed in the real-time instrument software. So the software does have delta CT built into it. But we also have free software that can be downloaded from our website that can have a few additional features for group-based analysis that might be of interest. One is called DataAssist, and DataAssist may be, in fact, particularly useful for array type of experiments. Again, it's free software. The way this works is one would export the results from the real-time file and then bring that into DataAssist. And DataAssist offers a number of plots in addition to the RQ plot. There's graphs like heat maps, scatter plots, volcano plots. Could be interesting. We also have free software, does a similar thing called Expression Suite. And in this case, Expression Suite accepts the real-time files rather than the exported results. So that completes the presentation, and I thank everyone for participating in this. I'm going to bring up the chat. Let's see. Yeah, here we go. All right. So I'm bringing up the chat, and so if anyone has questions, I would say please go ahead and enter that into the chat. I'd be happy to answer. Oh, here we go. Okay, so the question is, which application in particular has Thermo Fisher applied this method, sorry, 5-nuclease assay? Is contamination an issue? Okay, all right. Well, that's a very interesting question. So we have many uh, real-time PCR applications, all using the 5-nuclease assay, or TACMAN. A lot of people like to call it that. We have gene expression is really popular. MicroRNA, of course, I've been talking about that in this presentation. We have copy number variation. We have a somatic cell mutation detection, SNP genotyping, um, fusion transcript assays. We even use, believe it or not, the 5-nuclease assay for quantifying protein, believe it or not. Those are called TACMAN protein assays. So yeah, we've been in. Might be useful also to point out that the 5-nuclease assay, or TACMAN, that was the very first real-time PCR chemistry, and it still is the gold standard. Next question, is contamination an issue? Well, it could happen to, that's not specific to TACMAN, of course, but contamination could affect just about any assay, I suppose. Generally, I would say that when it comes to diagnostic assays, contamination is a much greater concern than something like a research assay like gene expression. So as I said in one of the earlier slides, that with gene expression, we can tolerate a little bit of contamination. Generally, our scientists say 1% or less. Because you figure if you've got a 1% error, that's not going to affect your statistics. Like you're doing a t-test or something, that's not going to affect that. But in the case of diagnostics, you're looking for, let's say, a cancer gene or virus or something, any kind of contamination is not considered acceptable. So contamination, and it's not limited to real-time PCR either. Contamination can affect lots of different assays. So I don't know if the question relates to contamination specifically for the nuclease assay. I don't know if that's really the issue, or if it's two separate questions. But I can make a note that for gene expression, as an example, most of our gene expression assays position the TACMAN probe over exon-exon junction site. That prevents genomic DNA from being detected. So that's a form of contamination. If you're doing gene expression, you don't want to have genomic DNA as contributing to your results. So the TACMAN probe really helps in that regard. Keep that genomic DNA from participating in the data. Okay. Well, you're welcome. I appreciate the nice comment. Any other questions? We have a few more slides at the end. Okay. Next slide. There's the next
A few
slide.
more, yeah, a few more slides.
I didn't even realize there was a raffle. Okay, I didn't know about that.
Megan, did you want to come off mute?
Yeah. Hi, everyone. Thank you so much for attending. We wanted to just say if you were an attendee today, we will put your name into a raffle. and we will be drawing from that list. So stay tuned and see if you're the winner of the raffle. We have another slide. Let's see, get that advanced here. And we also have a promotion for you guys. See if we can get John to bring that up. I know we've had a couple audio issues. Well, if not, we can't get to the next slide. That's okay. I'll just go ahead and let everyone know we will be emailing out a promotional code. We're going to give you 30% off of microRNA assays. You guys can try this yourself. 35% off of TACMan Fast Advanced Master Mix, just to help you get you guys started in this process. And then we will also have our contact information available to you. So if you have any questions about this webinar or microRNA in general, we'd be happy to chat with you. And thank you so much for attending. We'll stick on the line a little bit longer in case some more questions come up.
Thank you. Stop the recording and I'll end the call.